Now, something I was mentioning a bit ago is that in prophase 1, and this is a real distinct characteristic, and that is the duplicated homologous chromosomes. They come together. We call that synapses. So they pair up. And what they do is they align themselves gene by gene. So the chromatids align themselves gene by gene. And when I say the chromatids, you have like a blue chromatid and a red chromatid pairing up gene by gene. And that will happen between the homologous pairs. As they do that, then what that forms is a tetrad, because that configuration, that synapse configuration, consists of 4 chromatids together. So we call that a tetrad. Now, a critical characteristic of prophase 1, and this is a characteristic you must remember about prophase 1, is that non-sister chromatids within a homologous pair will switch pieces with one another. We call that crossing over. And that is a critical event in prophase 1. What that does is that puts together new gene combinations so that the offspring will be genetically distinct or different from its parents. Now, another thing that's going on in prophase 1 is that there will be microtubules as the centrosomes continue moving to the poles, and they get to the poles. Some of the spindle fibers, some of the microtubule spindle fibers, will begin attaching to the kinetochores. Remember, the kinetochores are proteins at the centromere regions of chromosomes. And you'll have a microtubule attaching to a kinetochore of one of the homologs, and that will be from one pole. And then you'll have on the other homolog a microtubule from the opposite pole attaching. Now, what's going to happen is that's going to help the chromosomes move toward the metaphase plate, because we're going to have that whole tug-of-war thing going on between the opposite pole microtubules. Now remember, we call them kinetochore microtubules because they're attached to the kinetochores. Now, following prophase 1 and that whole tug-of-war thing, the tug-of-war leads to a stalemate, which places the homologous pairs on the metaphase plate. So they're essentially kind of lined up in the middle of the cell, but as they're lined up as pairs. Now this happens only in metaphase 1. The only time you will see homologous pairs of chromosomes aligned along the metaphase plate is metaphase 1. That happens in no other phase. That is the key unique component of metaphase 1. Now notice within the metaphase 1 drawing, we have a red chromosome on top, and then a blue, and then a red moving left to right. And then what do we have on the bottom? We have a blue, red, blue. So that's only one possible orientation for these 6 chromosomes. You could have had all red on top, blue on bottom. There could have been some other combination of red and blue on top and bottom. And the term for that is independent assortment. So anytime we use the term independent assortment, I need you to keep in mind that refers to the random orientation of homologous pairs of chromosomes at metaphase 1. Okay? So you don't always have the red on, say, the top and the blue on the bottom of the metaphase plate. That is not the case. That's one possible orientation. But there are others, and the orientation that these homologous pairs align themselves at metaphase 1 varies from one meiotic event to another. So anytime we use the term independent assortment, you now know it refers to the random orientation of the homologous chromosomes at metaphase 1. Okay, now following metaphase 1, we have anaphase 1. Now in anaphase 1, what we have motor proteins within the kinetochores, the little black buttons that we see here on these replicated chromosomes. Remember, the black button represents a kinetochore protein, and within that kinetochore protein is another protein called a motor protein. And what the motor protein does, it's kind of like Pac-Man. It starts chomping its way along the microtubule to which it's attached. Okay? Now we know that one of the homologs is attached to a microtubule, say in this case from the north centrosome, the top centrosome. The other homolog of that same pair is its kinetochore, its motor protein is attached to a microtubule microtubule that emanates from the bottom centrosome. And so by the motor proteins of the different kinetochores chomping their way along the microtubule, we get separation of sister chromatids. So oftentimes we'll say the motor protein is kind of like Pac-Man. It moves its way along the microtubule towards the centrosome pole, and that leads to separation of the homologous chromosomes. Now, one of the things we notice, though, is that the homologs still consist of two sister chromatids, so they're still in the replicated or duplicated state. Now, following anaphase 1, we have telophase 1 and also cytokinesis. Now, if you look at the telophase 1 drawing, you see that some key features is that the homologous chromosomes have the one set of chromosomes has made its way into essentially what's becoming a daughter cell, and the other set has made its way into what will become a daughter cell. So the chromosomes have arrived at the appropriate poles. There's been separation of the homologous pairs, and so each pole, which will become two different cells, has a haploid number of chromosomes. Now, if you notice within, say, this top telophase 1 cell, you can see the nuclear envelope beginning to build again. So a nuclear envelope will begin to build around this haploid nucleus, and the nuclear envelope will begin to build around the haploid nucleus within the other cell. So one of the key things that I want to point out is that, once again, within each nucleus of the two cells, there's one large, one medium, and one small chromosome. And so each nucleus, it contains a haploid set of duplicated chromosomes. And that's unique. You find that in no other phase other than telophase 1. Now, the other thing that's going on, we see, is that this animal cell is beginning to kind of pinch in a bit at the center. And we call that cytokinesis, which is when the cytoplasm divides. And so what we have by the end of telophase 1 and cytokinesis is we have two haploid daughter cells, and they are genetically different from one another. Look what we have. We have a blue, if we look at the top telophase 1 cell, we have a blue small chromosome with a red tip. We have a large red chromosome with a blue tip and with some blue tips and so on, and that's not what you see in the other cell. They're genetically different from one another because of independent assortment, metaphase 1, and crossing over in prophase 1. So two genetically distinct haploids. Now, we finished up with meiosis 1. We've separated the homologous chromosomes. Now we're going to run those two cells through meiosis 2. And in meiosis 2, remember, we said that we're going to separate the sister chromatids from one another. So we're going to move on into prophase 2, and we see fragmentation of the nuclear envelope. We see the mitotic spindle beginning to build. We see the centrosomes beginning to move apart, moving poleward. And so these are characteristics that are similar to what we saw back in prophase 1 and even prophase of mitosis. Okay? Now, another thing we're going to start to see are kinetochore microtubules actually aiding the movement of the chromosomes toward the metaphase plate. Now, a key thing, though, is to keep in mind that there will be no synapses, because synapsis occurs between homologs, and you only have one set of chromosomes in prophase 2, so there can't be synapses. And since there can't be synapses, there can't be any crossing over. Then we get into metaphase 2, because that whole tug-of-war thing that began in prophase 2 enabled the alignment of the homologous set of chromosomes along the metaphase plate. We can see, of course, the sister chromatids can dangle off of that metaphase plate, but we have the chromosomes lining up, single file at metaphase plate. There has been no independent assortment because we only have one set of chromosomes. We can't have independent assortment without homologous. Now we move on into anaphase 2. Now, when you look at anaphase 2, what we see, and this is the key event of anaphase 2, and that is we have one sister chromatid of a duplicated chromosome going to one pole of the cell, and the other sister chromatid is going to the opposite pole. So what we get in anaphase 2, separation of sister chromatids. And keep in mind, that's accomplished by way of the motor proteins that are contained within the kinetochores. So we get these sister. Following anaphase 2, we have telophase 2 and cytokinesis. Now, if we look at the drawing for telophase 2, we see that that one cell we began with back in prophase 2 is now splitting in half, and each of the two resulting cells are haploid, consisting of chromosomes that are not duplicated. So we have four cells, four resulting cells, and they're going to be genetically different from one another. That is certainly key to telophase 1, and of course we have the cells dividing, and of course that's what cytokinesis really refers to, as division of the cytoplasm. So this pinching in, this is what we call cleavage furrowing, is the result of cytokinesis, or we refer to it as being cytokinesis. Now, if we do a comparison of mitosis and meiosis, I think this will kind of help bring out some of the key distinctions that we have pointed to. So we have this theoretical cell, and its diploid number is 4, and that's what we have in the center part, at the upper center part of this drawing, is we have one large red, one large blue, one medium red, one medium blue. That's 4 chromosomes. They're in the non-replicated state. Now let's take this cell and let's go to, say, a somatic cell of this species. And what that species would do in a somatic cell, of course, is run that cell through S part of interphase so that each of the 4 chromosomes are replicated or duplicated, and then in the case of mitosis, eventually at metaphase, all 4 of those chromosomes will be lined up single file along that metaphase plate. Now let's compare that to what this species, this theoretical species, would be doing with those 4 chromosomes in meiosis in their gonadal tissue. All 4 chromosomes would be run through S part of interphase, and then look how they would line up in metaphase 1. They would line up as pairs, which is very different from what we get in metaphase. Now keep in mind too, a key distinction, a key difference would be back in prophase 1, synapses and crossing over. And keep in mind at metaphase 1, in this particular drawing, we have the 2 red chromosomes on the left, the 2 blue on the right, but there could be other configurations, and remember, we call that independent assortment. So anyway, we can compare metaphase and metaphase 1 and see that the way that the chromosomes align is very different. They line up as pairs on the metaphase plate in metaphase 1. They line up single file in metaphase. So what that allows then is that allows separation of homologous pairs by the end of meiosis 1, so that there are 2 resulting cells, each having a haploid set of duplicated chromosomes. Whereas in the case of mitosis, we get separation of sister chromatids, and that results in 2 daughter cells. And look at the 2 daughter cells genetically; they're going to be identical to one another. Whereas by the end of meiosis 1, the 2 resulting cells will be genetically different from one another. And once again, crossing over is key in that, as is independent assortment. And then, of course, we run the, in the case of the meiosis side, we run each of the 2 cells through meiosis 2. That leads to a total of 4 cells being produced. Each is haploid, and each is genetically different from one another. That is key in my. Okay, so we've been talking about independent assortment at metaphase 1 and crossing over. Helping organisms to produce varied offspring, genetically varied offspring. Random fertilization is a part of this as well. So, just a reminder, independent assortment occurs at metaphase 1. It's the random orientation of homologs. We see that being shown to us here in the 2 drawings, possibility A, possibility B. Notice that in possibility A we have the 2 blue chromosomes on the left, the 2 red on the right. That's one configuration at metaphase 1. We go to possibility B, and we can see a blue and a red on the left and a red and a blue on the right of the metaphase plate, and that's a second So that is independent assortment. Random orientation of the homologs at metaphase 1. Now, in the case of determining the number of orientations at metaphase 1, all you have to do is take 2 to the n power, where n is the number, the haploid number of chromosomes. So, for instance, here's a question. We'll see if we can kind of figure this out. How many different gamete combinations can a species that contains 4 chromosomes in the diploid state produce? Now remember, the number of combinations is going to be 2 to the nth power, and n is the haploid number. So what's the haploid number going to be for this species? Well, it's going to be 2. So 2 to the 2 squared to the 2 power equals 4. There are 4 possible arrangements at metaphase 1. What about if the 2n number for species is 10? Well, that means its n number would be 5. So we would take 2 to the 5th power. That means there are 32 possible arrangements at metaphase 1 of the homologous pairs in terms of red and blues lining up, lining up at metaphase 1. But what about humans? Well, we know our diploid number is 46, so that means our n number is 23. And if you take 2 to the 23rd power, that's some 8 million possible arrangements of our homologous pairs at metaphase 1. So whenever a gamete's produced, and we're looking at one gamete, that's only one of about 8 million possible combinations. So that's, if you come from a big family, and that's part of the reason that your siblings are going to be different from one another, is because of independent assortment. Now, another thing, we've talked about crossing over, and that's what's being shown in the colored diagram with the blue and the red chromosomes. And we can see that crossing over has occurred. And this is an actual electron micrograph in the center of the screen. And we've talked about crossing over occurring during prophase 1. They estimate that about 1 to 3 crossover events will occur per chromosome pair. Now, where you have this kind of X shape, where the 2 non-sister chromatids are switching pieces, that X shape is called a chiasma. And so you can see that even in the micrograph here in the center of the slide. You can see that X shape, and once again, the term for that is a chiasma, and that's where crossing over has actually occurred. Now, what we have, of course, in crossing over is that the 2 non-sister chromatids within the tetrad will break at the same place, and what they then do is they trade places with one another. And of course, what this does is it recombines genes in ways that's certainly different from, say, mom or dad. And we call these kinds of chromosomes that experience crossover events, we call them recombinant chromosomes. And if a chromosome doesn't experience a crossover event, then we refer to those kinds of chromosomes as being parental type chromosomes. So we can see here in the diagram on the right, they're showing a homologous pair of chromosomes with genes C and E. We can see in the second drawing, working our way down, there's a break. And then we have the 2 pieces actually being switched between the 2 homologs. And then once we get separation, you can see that leads to different gene combinations in a couple of the chromosomes. And then those, of course, are recombinant chromosomes, and then the sister chromatids, or the chromatids that become chromosomes that didn't experience crossing over are referred to as the parental type. Now, another component of offspring being different from the parents is that of random fertilization. So we said we can produce, lots of different gametes because of independent assortment and crossing over. And which 2 gametes are involved in the fertilization event? Well, that's a bit random. And so there's plenty of potential for offspring to be different from parents in sexual reproduction. And that's key for a species being able to adjust Now, there can also be accidents occurring during meiosis, and this can actually bring on a change in the chromosome number within gametes. And so what we're going to be looking at on this slide is a non-disjunction event during meiosis 1. And we're going to see that it's going to affect the chromosome number in the resulting gametes. Now, what we have in a non-disjunction event, and it's drawn here in meiosis 1, this drawing in meiosis 1 is really that of anaphase 1. We should get separation of homologous pairs, right? We've talked about that before. But look what's happening in the bottom pair. They're not separating. The mechanism to separate those homologous chromosomes has failed. And so what that's going to lead to is an abnormal number of chromosomes in the resulting gametes. Now, there's a term for that, and the term for that is aneuploidy. So a gamete having an abnormal number of chromosomes is aneuploidy. Now look what happens. So since we're having failure at meiosis 1 of separation of the bottom homologous pair, we go into meiosis 2 and we see that all the sister chromatids are being separated in anaphase 2, and we're having that with both of the cells. But then look what happens in the gametes. 2 of the 4 gametes will have an extra chromosome. 2 of the 4 gametes will be missing a chromosome. So those gametes having an extra chromosome are referred to as N+1 cells because they're N haploid plus 1 extra chromosome. Those gametes that are missing a chromosome are referred to as N-1. So an N+1 gamete has 1 more chromosome than the haploid number than it should. And N-1 Now, we can also have a non-disjunction event occurring during meiosis 2. Now, in the case of a non-disjunction event occurring during meiosis 2, we see that in meiosis 1 we get an appropriate anaphase 1. We get separation of the homologous pairs. When we get into meiosis 2, look what's happening at anaphase 2. We're not getting separation of sister chromatids for one of the chromosomes. Now, what does that lead to in the gametes? Well, we have one gamete that's an aneuploidy, it's an N+1. The other gamete resulting from this non-disjunction cell will be an N-1. But then look what's happening in the other cell. The other cell produces perfectly normal gametes. So there's a 50% probability that this individual will not ever know that they, had a non-disjunction event occurring in during the gamete production because, because half of the gametes will actually be normal. So half the gametes will be normal, 1/2 will be aneuploids if the non-disjunction event occurs in meiosis 2. So let's remember then the key difference between non-disjunction event meiosis 1 and meiosis 2, that if the non-disjunction occurs in meiosis 1, all 4 gametes will be aneuploids. If non-disjunction occurs during meiosis 2, then it'll be... Now, Down syndrome is caused by a non-disjunction event. The individual has a condition called trisomy 21, which is where they have an extra copy of chromosome 21. So if we look at the karyotype of this young lady, we see that she has 2 number 1 chromosomes, she has 2 number 2 chromosomes, of course one's from mom, one's from dad, 2 number 3s, 2 number 4s, all the way down, but look what's in the box. Look at her number 21 chromosome. She has 3 of those. So one of her parents probably experienced a non-disjunction event. There are other ways to develop Down syndrome, but experienced a non-disjunction event. Now, this is the most common aneuploidy condition. It affects, and the numbers vary a little bit according to one author to another, but the number that's given in this text anyway is about 1 in 700 individuals will have Down syndrome. And you think about that value, that seems really pretty high. It does have an impact on the individual. Lifespan is usually shorter than normal. It also, about half the eggs that a female with Down syndrome produces will actually have an extra chromosome number 21. Men who have Down syndrome are usually sexually underdeveloped and sterile. Now, one of the things that the graph provides for us over here is that there's a correlation anyway between increased risk of Down syndrome and increased maternal age. And so you can see that early on, in kind of the prime years having children, that the risk of having a Down syndrome individual is quite a bit less. But once you hit, say, I don't know where you want to pick a point, but somewhere around age of 40 on up, there's a dramatic rise in the risk of having a Down syndrome. So here's some other, in this table, we have some other, aneuploidy conditions. Here's Klinefelter's. Klinefelter's is an individual, male individual, and they actually have 2 X chromosomes and 1 Y. So they have an extra X chromosome. The frequency in the human population, or population here in the U.S., about 1 in 2,000. I've seen it at 1 in 1,800. So I mean, I've seen some different numbers, but somewhere around 1 in 2,000. There are also some males that have 2 Y chromosomes with their X instead of just the 1 Y chromosome. That happens about with the same frequency as Klinefelter's. There are some females that have 3 X chromosomes, but that doesn't have an impact on their phenotype. That's about 1 in 1,000 females in this country, so that's pretty high when you think about it. And in this XO, that really is a 0. This is a female who has 1 X chromosome. She's missing her second X chromosome, and that's called Turner syndrome. And that's about 1 in 5,000 females that have that condition. So I have a question for you, since we've talked a little bit about some of these non-disjunction events and different kinds of gametes that can be produced by that. Which kind of gamete would produce a Down syndrome individual? Would it be an N+1, or would it be an N-1 cell gamete, or an N gamete? And of course, what it would be is, it would be an N+1 and an N getting together, essentially. So I'm going to say N+1, just once again reminding us that there is 1 extra chromosome. It's 1 extra number 21 chromosome. Now, which non-disjunction event can produce a Down syndrome? A non-disjunction event occurring in meiosis 1 or meiosis 2, or either? And of course, the answer to that is going to be either. Now, another thing that can happen is that a change in chromosome structure can cause birth defects in individuals, and also even cancer. Now, one of the things we're going to talk about here at the beginning is something called a deletion. A deletion is when a chromosome fragment actually becomes detached. Now, you see that in the drawing here. It's highlighted in purple. The part of the chromosome that becomes detached, and it's lost. Now, the chromosome's repaired, but without that fragment. That's a deletion. And so it can in time be that chromosomal fragment actually just disappears from the cell. Now, another thing, though, that can happen is that fragment can become attached as an extra segment to its sister chromatid, or even to its homologous chromosome. In other words, the other chromosome that is genetically like it. Now, if that's the case, then that can lead to a duplication. So, for instance, we have the duplication being illustrated in this diagram, and we see this purple chromosomal fragment. It gets broken out, and then it gets added to a homolog. And that leads to a duplication for that particular chromosome. Sometimes the fragment is actually added back to the original chromosome, but in reverse orientation. And the term for that's an inversion. So there are all kinds of strange things that can happen with our chromosomes. Well, we've already mentioned that a fragment may very well join with a homologous chromosome, but also the fragment can actually be added to a non-homologous chromosome. Now, the term for that is translocation, and that's what's being shown down here between chromosome 9 and chromosome 22. Now, in this case, we're having what's called reciprocal translocation, and that's where a piece of 9 breaks off and a piece of 22 breaks off, and the two pieces get switched, and you can see that down here in the bottom drawing. And what happens in this case is that the 9 fragment added to the 22 places a gene close to what's called an active promoter. We'll get into that later. And this can lead to something called chronic myelogenous leukemia. And so it actually becomes a cancer-causing gene, because it's now been placed next to a very active promoter. And this is called the Philadelphia chromosome. So there can be reciprocal translocations as well. where fragments of two non-homologous chromosomes actually switch places with one another. There can be other issues as well. There's something called ch8. Now this is where an individual experiences a deletion in chromosome 5. And a deletion can be critical, certainly if it's affecting, genes on the chromosome. And it can, in the case of ch8, the deletion is great enough that it can lead to severe developmental disabilities. And the reason it's called ch8, that really stands for cry of the cat, but that's because their cry, the baby's cry, actually sounds like the mewing of a distressed cat. Now, these individuals, it's not uncommon for them to die in infancy or early on in childhood. I mentioned the reciprocal translocation that can lead to chronic myelogenous leukemia, and that's what we have over here in this diagram. And once again, that's a reciprocal translocation between chromosomes 9 and 22, and so this once again leads to an overactive gene that's involved in cell division.